All right, well, hello everyone, and welcome to Anatomy 152 Exam 4 review. What I've done is similar to what I've done in the past. I've compiled all of the review guides / study guides that are, you can already find those on Canvas, with all of the images and PowerPoint that I feel to be relevant. So you'll note that there are some images that are not present, and that's because it's something that is not going to be tested or not strongly leaned upon. So I'm just going to go ahead and hop right in here. So we're going to start with the metabolism and nutrition outline. And so I have here, as I mentioned, all of the information that's going to be in the study guide as well as the images. So metabolism is basically all of the chemical reactions that occur in the body. And these are all going to be reactions that are either going to require ATP, which means that they're going to need to have energy put in, and ATP is going to provide that energy. Or it's going to be something that's going to be exergonic reaction, where we're going to have energy released, and that energy is going to be stored in an ADP and a free phosphate becoming ATP. So this ADP and ATP cycle that you're seeing here is basically showing you how certain reactions, namely reactions that build things up, right, anabolic reactions, are going to require energy put in, and reactions that break things down are going to release energy that's going to then form ATP. Right, so that's what you're seeing here in this pretty little diagram. All right, so we talked about reactions being one of the major ones that we'll see quite often is a redox reaction or an oxidation reduction reaction. Here you go. And so here we have an example of oxidation reduction reactions. And the oxidation is basically going to be when we are removing hydrogen, or removing electrons, right? And that's going to mean that we're going to lose the hydrogen atoms. So an oxidation reaction can also be called dehydrogenation reactions, which you probably won't see very often. And then a redox reaction would be the opposite. So if lactic acid going to pyruvic acid is the oxidation reaction, the opposite reaction, so going from pyruvic acid back to lactic acid, is going to be a reduction reaction, right? So you're always going to have them equal and opposite. And also they're pretty much going to be depicted as redox reactions simply because every time we have a reduction reaction, there's also, at the same time, typically, an oxidation reaction. The next thing down here is showing NAD and FAD. NAD and FAD are able to become reduced to NADH and FADH. And by reduced, I mean obviously they picked up the hydrogen, which means that they've also picked up the electrons. And that's going to be another energy carrier molecule that we use in the cell, similar to but not equivalent to ATP. As I mentioned, like we had $5 bills and $20 bills. ATP is our main carrier molecule, but we also use FADH and NADH. And then when we look at the reaction of when we break down glucose, right? So respiration, we end up with NADH and FADH also popped out from these cycles, which then towards the end are going to get converted into ATP when they enter into like the electron transport chain. Goodness gracious. All right, here we go. So for you guys, oh goodness gracious. All right, so this is basically just showing you that we have the opposite reaction can occur as well. So if we have lactic acid going back to pyruvic acid, that would be an oxidation reaction. But if we have pyruvic acid going to lactic acid, that's a reduction reaction. And that they're always coupled. So that's also the point here, is that when we are having an oxidation reaction, in this case we're losing hydrogen ions from the pyruvic, from what's becoming pyruvic acid, right? Lactic acid loses the hydrogen and becomes pyruvic acid. That's going to be coupled with a reduction reaction where NAD plus is acquiring that hydrogen to become NADH. So we have always an oxidation and a reduction reaction occurring at the same time, which is why you pretty much always hear of them called redox reactions, right? Because they're both happening at the same time. And then the next one is just showing you ATP generation, and at this point you should pretty much know that ATP is created from ADP plus a free phosphate and energy. So when ATP loses its phosphate, energy is released, and it goes down to ADP and a free phosphate, depicted here. When we have energy put into the system, this free phosphate is going to get linked to the GP molecule, becoming a triphosphate molecule, which is the high energy form. And you can see this little squiggly bond, and that's going to basically designate a high energy bond, right? And then the next thing we're going to talk about here is carbohydrate metabolism or sugar metabolism, which is broken up into 3 parts. The major part that we're going to see first is going to be glycolysis, and then we're going to see the Krebs cycle, and then we're going to see the electron transport train. Okay, so glycolysis is the very first thing that happens, and glycolysis is anaerobic. That means that it does not require oxygen to occur. And in the absence of oxygen, this is as far as we get. We go from glucose to 2 pyruvic acids. We take 1 6-carbon molecule, break it into 2 3-carbon molecules. That's as far as we get if we don't have oxygen present. If we do have oxygen present, from there, pyruvic acid gets converted into acetyl coenzyme A. Again, in the absence of oxygen, pyruvic acid gets converted into lactic acid, which is what builds up in your legs when you get cramps because you don't have enough oxygen to be able to turn over all the pyruvic acid. So in a pinch, breaking down glucose into 2 pyruvic acid is still going to give you energy, right? It's still going to give you 2 ATP and 2 NADH. But that small change in comparison to what you're going to get, if you then take that pyruvic acid, send it through the Krebs cycle, and through the electron transport chain. And so this little diagram here is showing you how much energy you get out at each of the major steps as an overview. And of course, we're going to go into it in more detail. So glucose has a lot of different options in terms of what it can become. So glucose can be, glucose that's absorbed from the diet can do a couple of things. One, if we need energy immediately, it can be utilized for energy immediately, right? We can also use it to form amino acids. I'm going to make this just a little bit bigger for you guys. We can also use it to form amino acids, which we can then incorporate into proteins. Or we can store the excess glucose in the liver or skeletal muscles in the form of glycogen. And when we're creating glycogen, that's called glycogenesis, right? And we can also take that glycogen or the glucose, which would be converted into glycogen, and convert it into glycerol and fatty acids that we can use to create fat, basically, triglycerides, which was called lipogenesis, or the creation of lipids. Okay, so glucose is going to get picked up through sodium glucose symporters and then get transported into the blood, from the blood to the body cells, and then gets into the body cells through facilitated diffusion, specialty types of diffusion transporters called GlyT molecules. And as we talked about insulin previously in regulation of blood glucose levels, we talked about how insulin helps with the uptake of glucose. It does that by inserting those diffusion transporters into the plasma membrane, which facilitates the uptake of glucose into the cells, right? And once it goes into the cells, it's going to have a little phosphorylation event that occurs. Once that phosphorylation event occurs, that glucose becomes trapped within the cell. At that point, it's going to be targeted for cellular respiration. Okay, so that's also going to be called, where are we? Glucose metabolism, one more. There you go. That's going to be called cellular respiration. It happens in all of the body, with the exception of red blood cells. Red blood cells don't have mitochondria, so they're not able to undergo this process because, remember, mitochondria are going to take it from pyruvate on. It's going to enter into the mitochondria. And this is going to create a large amount of energy for the cell. We have 4 major stages that are going to happen. First, glycolysis that we talked about, and then formation of acetyl coenzyme A, which I kind of glossed over because that's really just pyruvic acid getting converted. But then from there, we go into the Krebs cycle, which has all of those major stepwise reactions where we're pulling energy out of those glucose molecules or those pyruvic acid molecules as we make small little conversions. And then after that, what's left is going to enter into the electron transport chain, whereby we're going to be able to get the rest of the energy out of it. So you guys, here we go. So the electron transport chain is where we're going to take all this extra NADH and FADH, all those smaller Vldls, and convert them back into the bigger currency like ATP, which is what we use most often. And so that's going to be the last stage here, that electron transport chain. All right, so now that I've told you what I'm going to tell you, I'll tell you again. Glycolysis is when we break down that 6-carbon molecule, which is glucose, into 2 3-carbon molecules, that's pyruvic acid, and that's going to produce 4, which is a net gain of 2. And I want you to pay attention to this because there's a tricky question that asks you what your net gain is, and one of the answers is that it's a net gain of 4, which is not true. It's a net gain of 2, right? It's a total gain of 4. So make sure that you read the questions carefully, because we're bringing 4 out, but because we're putting 2 in, we're only getting 2 additional out than we started with, if that makes sense, right? So read your questions carefully. That one gets students all the time. That's my little winky face for those of you that came to the review. All right, so we gain 2 here, which is small change compared to what we're going to get, once we send that pyruvic acid into the mitochondria. And again, that only happens when we have oxygen. So as oxygen is in short supply, pyruvic acid is going to become lactic acid, which again is what builds up in your muscles and gives you that Charlie horse feeling. So normally, however, it's going to have oxygen, which means it's going to get popped into the mitochondria and enter into the Krebs cycle. First, it gets converted into acetyl coenzyme A, and once it's in the Krebs cycle, it's going to then create a lot more energy. So first, you have the formation of acetyl coenzyme A. All right, and again, as I mentioned, the fate of pyruvic acid depends entirely on whether or not oxygen is available. If oxygen is available, it enters into the mitochondria and enters into Krebs cycle, etc. If oxygen is not available, it becomes lactic acid and is going to end up just being destroyed in the body later on, so go down different pathways, right? That's going to be the end of how much energy we get out of it. However, since we do have oxygen available, we form acetyl coenzyme A, which then is going to be used in the mitochondrial matrix to enter into the Krebs cycle. And the Krebs cycle should be here. It should be here. All right, so let me make this a little bit smaller for those of you at home. Sorry, I have different screens on both of them. So the Krebs cycle is also known as the citric acid cycle. So you may know it as the citric acid cycle if you learned it when you were in high school. And it's basically going to be stepwise biochemical reactions that are going to happen again in the matrix of the mitochondria that are going to allow us to extract energy stepwise through the conversion of these molecules from one to the next. So there's going to be a lot of what we call decarboxylations. What's that mean? We're pulling off a carbon, right? So we're going to have multiple steps. Decarboxylations, oxidations, and reductions of these different acids, which are stepwise going to be taking what was a 3-carbon molecule and eventually turn it into 3 single-carbon molecules, right? 3 carbon dioxide molecules. Excuse me. All right, so this shows you the simplified and the more complicated version of the Krebs cycle. Basically, what's going to happen in the Krebs cycle, and as I mentioned, I don't expect you to memorize every single step of the way, but you will need to know what steps are what. So I need you to know that we start off with citric acid and go to isocitric, etc. So yes, you do need to know which ones go in where, but you don't need to know any of the biochemical structures of anything or which conversions happen that's a phosphorylation or where does that carbon get broken off, etc. But what I want you to know is what goes in, right, which in this case is going to be acetyl coenzyme A. Coenzyme A also almost immediately comes off, so it's going to then get recycled to get back into the conversion of pyruvic acid into acetyl coenzyme A, right? So that coenzyme A just constantly goes through a cycle. That leaves us with citric acid, which is why citric acid is what the name of this cycle is or was when we were in high school. From citric acid, we go to isocitric acid, just a quick little isomeric conversion. And then we go to alpha-ketogluteric acid. At this point, from isocitric acid to alpha-ketogluteric acid, we are creating one NADH molecule. And then we create another one as we go from alpha-ketogluteric to succinyl-CoA. Right, and again, this is another place where coenzyme A kind of rotates through just for 1 second. It comes in, becomes acetyl coenzyme A, and then pops off again as we go to succinic acid. And that's going to help create ATP by creating, for a very small period of time, GTP first, and then ATP converted from there. So at this point, we have released 2 NADHs and 1 ATP. We're going to release 1 more FADH and another NADH before this is over. And then the very final product here, which is oxaloacetic acid, is going to be used to convert the acetyl coenzyme A into citric acid. So the byproduct, the very end product, enters back into the pathway, and that's why it's called a cycle. And every time that we have the acetyl enzyme CoA come in, so every 2, we're going to end up with 6 NADH coming out, 6 hydrogen, and 2 FADH2. And again, all of these guys are then going to get converted into ATP when they enter into the electron transport chain. All right, so at the electron transport chain level, which is here, we have a sequence of reactions that are electron carrier molecules that start on the inner mitochondrial membrane and pop things across to the outer mitochondrial membrane. So we have a series of oxidation reduction reactions that occur whereby the hydrogen ions are getting pumped out and then they're getting pulled back in. So the hydrogen ions come off of that NADH, FADH, etc. Pardon me. Get released into the lumen, and then, into the area between that outer membrane and the inner membrane. And then they're going to come back in through ATP synthase, which looks kind of like a child's slide at an amusement park, right? A little swirly slide. And as that comes back on through, it's going to drive the creation of ATP. So we're basically changing out our currency from 5s back to 20s, right? From NADH and FADH into ATPs. So that's essentially what we are doing here when we're talking about the electron transport chain. So as the electrons get passed through the chain, there's a stepwise reaction where the energy is released stepwise to help create ATP. And again, this is going to require aerobic respiration, right? It's going to require oxygen because the very last electron receptor is O2. And so that very last step is going to be irreversible. So at that point, we're going to have that the electron lost from this chain. All right. So to summarize cellular respiration, we've got this pretty little figure here. We're going to start off with glucose, that's C6H12O6, which is written oddly here, but it's written correctly in the actual image. The superscript and subscript are strange in Word. But in the image, this is what it should look like, C6H12O6. And then we're also going to be bringing in oxygen, right? Where does that oxygen come from? Well, the process of respiration, actually ventilating the body. And then that's going to be delivered by blood to the tissues. So the tissues are going to receive glucose and oxygen from the blood supply. And then they're going to use that to convert that glucose stepwise into carbon dioxide, water, and ATPs. And also depicted here are the ADPs and the free phosphates that you're going to need to be able to have that come out, right? Because in order to get those 32 ATPs, they have to come from somewhere. They come from ADPs and free phosphates. So essentially what you're doing is you are stepwise taking a 6-carbon sugar and breaking it into individual carbon dioxide molecules, releasing energy throughout the way that's going to get converted into either directly into ATP or into FAD and NADH. FADH and NADH would then get converted into ATP at the end, right? And so that's aerobic respiration. And aerobic respiration can create 36 or 38 ATPs from each molecule of glucose, right? And so that means that we can actually have a net gain because we're going to have put those, first we put some in, and then we also are going to have gotten some out of the breakdown of glucose into pyruvic acid gives you 2. So only some of this is going to come out of the aerobic respiration pathway, but when I say some of it, I mean most of it. Otherwise, we would have gotten a net gain of 2. Here we have a net gain of between 30 to 32. All right, so these are all of the major events that occur in cellular respiration. All right, so basically what happens is, sorry if you guys at home, I can't quite show it all. Basically what happens is sugar comes in and enters in the process of glycolysis. This does not require oxygen. Then that is going to turn into pyruvic acid. That's our 3-carbon sugar. Well, it's not a sugar, a 3-carbon molecule. That's then going to enter into the mitochondria. Now we need oxygen. At this point, we're going into the aerobic pathway. That then enters or becomes acetylcoenzyme A, which enters into the Krebs cycle, stepwise series of reactions, creating a little bit of ATP, but also NADH and FADH, which eventually is then going to get converted into ATP via the electron transport chain, resulting in the release of what? Carbon dioxide, water, and energy. So what do we start with? Sugar and oxygen. What do we end up with? Water and carbon dioxide. Now I'm going to take a step back here. What did the plants start with during the process of photosynthesis? Water and carbon dioxide. So photosynthesis and cellular respiration are equal and opposite pathways. One is taking the energy from the sun and harnessing it in the high energy bonds of glucose, photosynthesis. And the other is taking those glucose molecules and breaking them down and releasing the energy from those high energy bonds to be able to drive cellular metabolism, right? And we use that to create ATP every time that we release that energy. All right. So glucose has a couple different fates within the cell, as I mentioned. So it can enter into these pathways, right? These glycolysis pathways, or it can be converted into glycogen for storage, right? And so this is the image of how we're going to convert it into glycogen for storage, which is going to get converted into glycogen for storage in the muscle or in the liver. And so there's a couple of stages in this pathway. Basically, it's going to enter into glucose 6-phosphate, glucose 1-phosphate, and then eventually into glycogen. And then the opposite pathway can also happen if we need to take glycogen and convert it into sugar for the body to use, right? And that's called glycogenolysis, breaking down glycogen. So this is glycogenesis, creating glycogen. Glycogenolysis is going this direction for those of you at home. Glycogenesis, glycogenolysis. And in this way, glucose and glycogen can get converted back and forth, so we can help regulate our blood sugar. Even if we've taken in a large amount of it, we can store the excess in the form of glycogen. If we have too much glycogen, then we can store it in the form of triglycerides. All right. So if you're going to need to have a lot of energy available to you, for example, you're a marathon runner or something, you can do something called carbohydrate loading or carb loading. And if you eat a large amount of complex carbohydrates, that means that those carbohydrates are going to slowly break down into sugars so that you can have an energy source for a longer period of time. So you have a lot of energy available for your exercise. The other term that I haven't talked about here, I think I talked about it earlier, gluconeogenesis, or the creation of new glucose molecules. And that can happen from either protein molecules, fat molecules, etc. So we have a lot of crosstalk in these molecular pathways, whereby if we don't have one energy source, we can convert to another, which can then be utilized for energy. So several different compounds can enter into the Krebs cycle to provide energy, including certain amino acids are able. So glycerol can be converted into glyceraldehyde 3-phosphate, and specialty amino acids can also be converted into pyruvic acid. And in this way, by becoming pyruvic acid, or being converted into pyruvic acid, they can enter into the Krebs cycle, just like sugar can. Okay. Sorry for those of you at home, I apparently lost my place. Here we go. So lipid metabolism. All right, so remember, lipids aren't necessarily going to be able to go directly into the blood supply. Lipids are going to have carrier molecules that bring them into the blood. So we have specialty lipoproteins. So this is an example of these lipoproteins. We have specialty lipoproteins that are called chylomicrons, which allow us to transport dietary lipids across. We also have very low-density lipoproteins, or VLDLs. We have low-density lipoproteins and high-density lipoproteins. And so this is what we think of when you think of your cholesterol, right? Your LDL and your HDL levels. L being for lousy, so you want to have a higher ratio of HDLs and a lower number of LDLs, so you want to have high HDL. And that's because these guys are going to be responsible for delivering cholesterol to the tissues or taking cholesterol from the tissues back to the liver for excretion. And so since LDL takes it to the tissues and HDL takes it away from the tissues, you want to make sure that you have enough carrier molecules pulling the cholesterol back out of the tissues so it doesn't build up, right? And so that's what they do when they look at your cholesterol levels. They look at your LDL and your HDL levels, and which can give them kind of a thumbprint as to how well you're doing in terms of your cholesterol metabolism. All right, so this here is a micelle. You can basically see that we have polar heads and nonpolar tails on these little phospholipid molecules. These are the same phospholipids that you would see in your plasma membrane. Of course, the plasma membrane is a bilayer, so you would see two of them. And embedded in this membranous structure, you have ApoE, ApoC2, and ApoB100. These are all different types of lipoproteins that can be embedded inside these micellular structures. All right, so as I mentioned, the chylomicrons are going to be found in your intestinal cells. These are going to allow you to pick up dietary lipids, which are then going to enter into the lacteals. Remember, they're not going to be able to enter directly into the blood supply, but they'll be carried into from the systemic circulation, once it gets into systemic circulation through the liver, into the adipose tissue, where it's going to get stored in adipocytes, or it can be used for muscle cells for ATP production. We also, like I said, have VLDLs and HDLs. VLDLs are going to have triglycerides in there. And basically, they're going to be transport vehicles that are going to take the triglycerides to adipocytes for storage. And these triglycerides were originally created in the hepatocytes in the liver. And the VLDLs, after we're dropping off the triglycerides, they're going to get slowly converted into LDLs. LDLs are going to be the ones that take the cholesterol and deliver it to the cells. And if we have too much cholesterol or too much of these LDLs, then we're going to be depositing cholesterol in the smooth muscle fibers of the arteries. And this, obviously, is no bueno. HDL is going to do the opposite. It's going to take cholesterol away from the body cells and transport it to the liver, where it's going to be eliminated. All right, so we have two major sources of cholesterol. We have liver synthesis, so the liver is constantly synthesizing cholesterol, and we're also going to be eating cholesterol. So we want to pay attention to our dietary cholesterol, because we can't really control how much cholesterol the liver is synthesizing. And we want to try to keep our blood cholesterol under 200 total, and 130 or less for LDL, and 40 or more for HDL. Okay, and if we end up with bad cholesterol levels, then we can treat that with diet, exercise, drugs, etc. All right, so lipids. Move along. Lipids can be split into fatty acids and glycerol, and this is called lipolysis, or breaking them apart. And that can help us to enter into catabolism of our fats. We also know that we can break these guys down to make acetyl coenzyme A, which will help break into the Krebs cycle. So we can actually use fats to enter into the Krebs cycle for energy production. In fact, fats are going to give us 9 calories per gram, whereas proteins and carbohydrates are going to give us about 3. So triglycerides can become fatty acids, which can enter into the cycle. We're also going to see that amino acids are also able to enter into the Krebs cycle. What does this mean? This means that we can use multiple different energy sources for ATP production, which is obviously going to be important because not only are we going to have a varied diet of different energy sources, but we might have feast and famine within our lifetime where we might have to rely on the energy the reserves of the body. Okay, so as I mentioned, proteins can also be broken down into their amino acids, which then can get hydrolyzed and enter into this cycle as well. So the proteins can be catabolized to enter into the Krebs cycle, and there's several different steps that have to happen. First, we have to have deamination, so taking out the amino group. We have to decarboxylation, taking out the carboxylic acid group, and hydrogenation, right, adding more hydrogens. But eventually, we're going to be able to convert these amino acids into a couple different things that are able to enter into the Krebs cycle. It can be converted into glucose, which can go in, fatty acids, which can go in, and ketone bodies, all of which are able to be processed by the Krebs cycle to create ATP. So that's not the general fate of proteins. Usually the proteins are going to be broken down into amino acids and then used to reconstruct more proteins. So usually we're going to break down proteins into the very smallest building blocks or small Legos and use them to make larger protein chains. But again, if we enter into a situation where we don't have sugar or fats, we can break down our proteins as well. So we have protein catabolism, which is breaking down proteins, and protein anabolism, which is creating more proteins. Remember, anabolic steroids pump you up and knock everything off the table and break them. So catabolism is breaking and anabolic is building up. Protein synthesis we talked about extensively, and it's kind of a complicated diagram, and I'm going to brush through this relatively quickly because we don't have a ton of time. Basically what happens is one amino acid at a time is brought in by a tRNA molecule. tRNA molecules have an anticodon region, which is going to also be correlated to what amino acid they carry, as well as whether or not it matches the codon region on the mRNA. So the mRNA is encapsulated in a structure called a ribosome. We have a small and a large ribosomal subunit. Here's our small on the bottom, here's our large on the top. The large has 3 sites: an entry site, an attachment site, and an exit site. They are known as A, P, and E. Not for any reason that I can tell. So A is where it comes in, P is where we have, processing, and E is exit. A for anticodon. So A is where the anticodon binding site is going to happen, where the anticodon on the tRNA binds to the mRNA codon, and then it's going to shift over into the P site. When it does so, it's going to acquire the amino acid from the previous tRNA, and that tRNA that has just let go of that amino acid is then going to exit out. The whole thing shifts over again, a new tRNA comes in, and in this way we get a longer developing protein molecule by adding one amino acid at a time to a developing chain. Unfortunately, that's really all the time I have to discuss it. That's an entire lecture. I'm sure that you will encounter it, though, later on in your biological career. All right, so this image here is depicting the idea that we have metabolic crossroads. What does that mean? That means that we have different ways that these molecules can go down different metabolic pathways. For example, glucose 6-phosphate is a very important one because it can do a couple of things. It can get converted into glycogen. It can get converted into glucose. It can get converted, excuse me. It can get converted into DNA and RNA, as well as getting converted into pyruvic acid. And pyruvic acid, as we all know, can enter into the Krebs cycle. So there's a lot of different ways that this molecule can be utilized by the body, depending on which areas are deficient. Right? So if we don't have enough glycogen, it can go that way. If we don't have enough glucose, it can go to the blood glucose. If we need DNA and RNA, right? Or if we need energy. So in this way, these particular molecules are very important because they serve as crossroad molecules that can enter into different pathways depending on the needs of the body. Okay, so some examples of that are going to include glucose 6-phosphate, pyruvic acid, acetyl coenzyme A. What's going on? All right, so we have metabolism during the absorptive state, which is here. So we have absorptive states and the post-absorptive state. The absorptive state is when we are actively absorbing food during digestion. What does that mean? That means our blood glucose levels are rising steadily. We can't have them rise too far, right? Our blood sugar has to stay within certain parameters. So what do? Well, we employ insulin to assist us, but basically what happens is glucose is going to get converted to a lot of different things. Glucose can get converted to glycogen stored in the skeletal muscle or in the liver, right? We can also take glucose and run it through tissues and oxidize it in cellular respiration and get energy out of it, right? Resulting in water, carbon dioxide, and ATP production. Or we can store glucose in the form of converting it into triglycerides in our adipose tissue. All of this serves to get the glucose back out of our bloodstream. Why? Because we can't have our blood glucose go too high. Just like anything, we need homeostasis. Plus, if you've got food, you may as well bring it in, right? If there's groceries on your step, you're going to bring them into your cell. So this is the absorptive stage. During this time, cells are creating ATP. We're oxidizing glucose quickly. We're also converting glucose into glycogen or to triglycerides. Additionally, we're storing our dietary lipids in our adipose tissue as well. And we're starting to convert our amino acids into carbohydrates, fats, or proteins, right? Whatever it is that we need for the body. So this is the absorptive stage. As we enter into the post-absorptive stage, boom, that's here. During the post-absorptive stage, we've already absorbed all of the glucose, etc. So our blood sugar is not rising anymore. In fact, it might be falling if we've already been pulling it out very quickly. So we might need to bring it back in. So how do we do that? So energy needs of the body have to be satisfied by what's already found in the body because nothing's coming in from the digestive tract. And so in order to maintain a normal blood glucose level, we have to take glucose back out of our skeletal muscle, which means we have to take glycogen and convert it back over, right? We also can have glucose coming out of, like I said, our liver, or we can have triglycerides being broken down into glycerol and fatty acids, which can be used to drive glucose creation in the liver. All of this is to help maintain homeostatic levels of glucose when we no longer have glucose coming in from the food. Oh, goodness, sorry. All right, guys, in the interest of time, I think we should be moving on to the next. So I'm just going to kind of scroll through until we enter. And I'm going to talk about nutrition. So the urinary system here. Just because I don't want to end up not making it into the last section. All right, what's going on? Where's my image? Well, you guys have it. Well, hopefully you can pull it up. Basically, the urinary system consists of kidneys. Each kidney is connected to a ureter. The ureters are both connected to a bladder. So we have 2 kidneys, 2 ureters, 1 urinary bladder, and then 1 urethra for excretion, which is how it's going to be expelled out of the body. Sorry that's not showing for those of you here. Nothing is showing for you guys anymore, huh? All right, so the organs of the kidneys are going to be there in the retroperitoneal area. They're a paired organ, which means there's 2 of them. We have a region called the Hillis, which is where... Are you guys able to see images on your side? There we go. Sorry, we just had to wait for it to catch up, I guess. All right, so I apologize, those of you at home. I know you can see images, but we were just caught up in class here. So we have a region that's a concave region of the kidney that has a little fissure in it called a hillis. And the hillis, we'll see this again in multiple different places. But the hillis is basically going to be a region where things come in or exit. In this case, it's going to be blood vessels entering, nervous tissue entering, and lymphatic tissue entering. And we have multiple layers around the kidney. If we go from the innermost region, that's the renal capsule. Then we have the adipose capsule, and then the outer renal fascia. Internally, we have a cortex, which is this region outside, the outermost region of the internal section. Then we have the medulla. The medulla is going to have pyramids in it. So, it's going to be surrounding the pyramids. So here's the medulla, and the pyramids are going to sit within that region. We also have papillae, columns, calyces, right, minor, major, and then the renal pelvis, pelvis for plural. And this is where the urine is going to drain out. The urine drains out from a couple different ways. It drains out by osmotic pressure, it drains out by gravity, and also drains out by peristalsis, or movement forward. All right. Next. Oh, I guess you guys are still seeing this. All right, so this here is showing you the path which urine takes. The urine is going to drain from the collecting ducts, which are going to enter into the renal pyramids. Again, these are going to be found in the renal medulla region. So renal medulla, pyramids found within there. We're going to have drainage that occurs through the collecting ducts into the papillary ducts, into the minor calyx, the major calyx, then into the renal pelvis, pelvis plural, out through the ureter, and then from the ureter into the urinary bladder. Now, the way that we're going to be able to concentrate urine is a specialty region called the nephron. The nephron is... I'm so sorry. I got my COVID shot and everything. All right, so the nephron is the most functional unit of the kidney. Basically what it does is it starts to concentrate urine, and it does this by having a really nice network of capillary bed across it, so anything that's going to be lost is going to get immediately picked up by the capillaries. And it enters from the cortex into the medulla, and as I may have mentioned last time, this is like crossing state lines, kind of, or crossing from Canada into the United States. There's different rules, and when I say rules, I mean there's a gradient of different concentrations of things inside the pyramids, inside the medulla, that is not found in the renal cortex. And so as we cross, we're going to enter into a gradient which allows things to escape, like solutes, and for water to follow as well. So this is everything that we're seeing here. First, we're going to have the blood filtered into this region here, which is a glomerulus, or Bowman's capsule. The glomerulus would be the side that has the blood supply. We have filtration occurring across there. We'll talk about that, how that works in just a second. But basically, it's going to get caught in this capsule and then enter into the proximal convoluted tubules. Remember, we've got capillaries everywhere, so we've got peritubular capillaries that wrap around this whole thing. From that region, right, this proximal convoluted tubule, it's going to enter into the loop. That's the descending limb. And then the ascending limb of the nephron loop. As we come back up, then we're going to enter into the distal convoluted tubule. From there, then we head back into the collecting duct, the papillary duct, and then out into the minor calyx. All right, so that's how urine is created. Basically, it's going to come out of the blood supply through a filtration process and then get concentrated through these loops. So we have the flow of fluid through the cortical nephron. Basically, we're going to have fluid coming in through the Bowman's capsule, then getting collected in the proximal convoluted tubule, then the descending and the ascending limb of the nephron loop, up into the distal convoluted tubule, which is then going to drain into the collecting duct, and then out into the papillary duct, into the minor calyx. Oh, goodness gracious, sorry. I'm missing one. Here we go. All right, so the nephron loop has multiple regions, including the descending limb, the ascending limb, and the distal convoluted tubule, right? I'm sorry, proximal convoluted nephron loop, distal convoluted tubule. And the nephron loop itself has, again, the descending limb, ascending limb, and then a thicker ascending limb. So we have two different sections, a thin ascending limb and a thick ascending limb. And we have two different types of nephrons. We have a shorter loop and a much deeper loop. Okay, so we have a cortical membrane, which is a much shorter loop, cortical nephron, sorry, which does enter into the renal medulla, but only a little ways. And then we have a juxtamedullary nephron, which runs much, much further deep, right? And is going to be a little bit different because it stretches all the way through the medulla and reaches almost down to the renal papillae, really, really far in. And these are going to serve different purposes in terms of the creation of urine, and we'll talk about that in just a little bit. All right, so next let's talk about the glomerular capsule. which is here. All right, so again, fluid is going to come in from the blood supply. It does that first from the afferent supply and then out through the efferent arterial. Blood's going to enter into this Bowman's capsule in the glomerulus, which is covered by podocytes. They have a visceral layer that's covered by these podocytes, and podocytes have little slits in them. So fluid can come out. Blood cannot, like blood cells cannot, but fluid from the blood supply can. And then that's going to end up exiting out into the proximal convoluted tubule from that subcapsular space, right? And then if we look at it a little bit more closely down here, this is what a glomerular endothelial cell looks like. So first, we have fenestrations or tons of pores on that endothelial cell, but we also have a basal lamina, so larger proteins can't come through. Then we have these slit membrane between pedicels. So the pedicels have these little slit membranes. So basically anything that gets out through this basal lamina membrane is going to be able to be secreted. That means the only things we're going to be keeping in, red blood cells themselves, of course, and larger proteins, larger and globular proteins who won't make it through these little holes. All right, so fluid is going to get filtered into the capsular space, and then it's going to end up coming out in the form of glomerular filtrate in the renal tube. And during this time, we're going to have resorption from fluid into the blood and also secretion from the blood into the fluid. So we're going to have back and forth between the blood supply and the urine. That's why we have all of these capillaries around this region. All right, and eventually, the blood's going to contain everything that we want to resorb, and the urine's going to contain everything that we want to secrete. All right, here we go. So we have this filtration membrane, which is a particular unit of the nephron that's found in that endothelial capsular membrane, and this is going to consist of, as I mentioned, the glomerular epithelium, the basement membrane, and then slit membranes between the pedicels of the podocytes, which is going to allow for fluid to go out. Then the filtered substance is going to move back and forth from the bloodstream into this urine, right? So we have things that are going to be picked up. But in the glomerulus itself, it has to pass through those three major barriers. First, the endothelial cell itself, which is a glomerular endothelial cell, which has a lot of little holes in it, or fenestrations. Then the basal lamina, but anything makes it out through the basal lamina is probably going to make it through that filtration slit as well. In fact, the only thing that filtration slit is going to keep in would be a blood cell. So what's the point? The point is to be able to force fluids through this membrane so that we can take it and release anything that we want to secrete from the body and resorb anything that we want to keep. And all of this relies on pressure. So we have what's called net filtration pressure, which is going to be a combination of multiple things. So first we have the glomerular blood hydrostatic pressure. This is how much pressure is coming in. In this case, it's about 55 mercury. We also have capsular pressure, which is how much pressure is inside the capsule already. In order for something to get out into that into that capsular space, it's going to have to go across these cells, right? And that means it's going to have to fight against the pressure that's already there. So what we are going to be looking at is the subtraction of what comes in, which in this case is 55, minus what's pushing back on it, including the pressure that's already inside the capsule and the pressure that the blood is preventing, that blood colloid osmotic pressure. So if we start with 55, we subtract 15, and then 30, we end up with 10, and that's what's going to be our filtration pressure. And this is actually something that we're able to change in response to how much water is in our body, et cetera. So we're actually able to hormonally, under nervous system control, have some sort of control over how much pressure is coming in to these regions. Okay, so let's talk about renal autoregulation. So we can autoregulate the glomerular filtration rate when we get information from receptors. Where are we? We get information from receptors from the juxtaglomerular apparatus, which in turn affects the afferent arteriole, which is where the blood supply is coming in. And we can change that afferent arteriole. In this case, we would constrict it, which would decrease the glomerular filtration rate. But we could also, if we wanted to increase that GFR, we could dilate it. Either way, we would return ourselves back to homeostasis by bringing that glomerular filtration rate back to normal. So as I mentioned, we actually do have some neuronal and hormonal control of these particular rates. Okay, so I just talked about the neuronal control. We also have hormonal regulation, which is going to be regulated through angiotensin II and ANP. All right, so we have multiple different resorption routes that a substance can take. It can either be resorbed directly across, such as here sodium, directly across the membrane into the capillary. That's by the process of diffusion. It can also be actively transported. This is the case of the sodium here, being actively transported into the cell, and then using a sodium-potassium pump can be pumped out. So it can move between the adjacent tubule cells directly or through the individual tubule cell by some sort of mechanism before entering into the peritubular capillary. And the fluid leakage back and forth between cells is called paracellular resorption. So as I mentioned, we have multiple different transport mechanisms. Yes. Here we go. Okay, so multiple different transport mechanisms. We have symporter and antiporter mechanisms. So a symporter mechanism would be an example of what's happening here. There's a brush border microvilli enzyme. Basically what's happening is we are picking up both sodium and glucose. Again, a symporter would be picking up two items going the same direction. An antiporter, on the other hand, here we can see here, sodium one direction, hydrogen the other direction. So an antiporter would be allowing one guy in and one guy out, right? And here's another example of the antiporter section, except it's going to be, although we're still using the sodium hydrogen, we're going to be having different molecules coming in through this path. Oh, gosh, I'm so sorry, guys. All right, yeah, so for those of you at home, this is a symporter. And this one would be symporter, right, sodium and glucose. This would be an antiporter, sodium one way, hydrogen the other. All right, so once we enter into the proximal convoluted tubule, here we go. Let's have it. Once we enter the proximal convoluted tubule, we're also going to have resorption occurring of multiple different things, including chlorine, potassium, calcium, magnesium, urea. Some of these are going to be able to go by simple diffusion. Some of them can go by osmosis, namely water, obviously, is the only thing that undergo by osmosis. And then some things can also, mainly everything's going to happen by diffusion, but sometimes it can diffuse in between cells, and sometimes it can diffuse across two sets of membranes, like through the cell. Either way, what's happening is we're getting resorption into the peritubular capillary of things like chlorine, potassium, calcium, magnesium, and urea. All right, once we enter into the nephron loop, so sorry you guys are missing these images. Once we enter into the nephron loop, we also, what if I do this? I click here. Hold on. All right, perfect. Once we enter the nephron loop, we also have absorption occurring. In this case, we have that sodium, potassium chloride symporter, where we're bringing in sodium, potassium, and chloride, just like you'd say. And then we're going to transport those across the membrane, either by the sodium-potassium pump, or chloride can just kind of leak right on through. Other cations as well can just come right on through by diffusion. All of that's going to get picked up in this vasorectal region, in the thick region of the distal region of the nephron loop. And at this point, we're going to be returning a lot of the a lot of the potassium back into the tubular fluid. And we're going to be resorbing sodium and chloride. And so what's happening at this point is we're not going to be absorbing a lot of the water, although some of it, about 50%, is going to be resorbed in the descending limb. Little or none of it is going to be resorbed in that ascending limb. So no water here. We're resorbing some water here, but we're resorbing mainly ions here. And remember, where the ions go, the water will follow. So then we have resorption occurring in the distal collecting duct. And in the distal collecting duct, we have absorption of sodium and chlorine, which is going to continue through the sodium and the chlorine channels. And then this is going to serve as the major site where parathyroid hormone, remember PTH, we talked about being responsible for calcium levels. this is where we're going to start resorbing a lot of the calcium. And again, very little accompanying water is going to get resorbed there. Now, as we enter into the late distal convoluted tubule and the collecting duct, now we're going to have, so now we're on G, yeah. Now we're going to have sodium passing through as well. Sodium pumps are going to transport sodium across the basolateral membrane. We're also going to have potassium secreted through leakage channels to have it secreted in the urine. So potassium can end up leaving into the urine that way. And then principal cells are also going to secrete a variable amount of potassium. And so we have a constant pump where we have this sodium-potassium pump where we're constantly bringing potassium in. So potassium is going to stay high, even though we have some leakage channels. And we're able to regulate tubular resorption and secretion by 4 major hormones. And really, if I could stress anything from this series of stuff that we've talked about, this is what you're going to be, I don't want to say tested on, but this is what you should be understanding the most, is how we are regulating the resorption of water. So we have the renin-angiotensin-aldosterone system, where we have angiotensin 2. Remember that happens when we convert angiotensin 1 in the lungs. So we start off with angiotensin 1, it has to make it to the lungs, and then ACE converts it to angiotensin 2. Angiotensin 2 is responsible for blood volume regulation, so it's going to increase your blood volume and therefore increase your blood pressure. And it does that by retaining more water, right? So it's going to be a regulator of electron resorption, and when we resorb electrolytes, water will follow. And that's going to increase resorption of water in the collecting duct. We also have ADH, which is responsible for increasing water resorption by increasing the water permeability in particular cells, the principal cells. We also have ANP. ANP is going to do the opposite. It's going to inhibit water and electrolyte resorption, right? If we don't bring in more electrolytes, we don't bring in the water because the water follows the solutes. And we also have parathyroid hormone, which we've talked about previously, which is responsible for calcium resorption. Okay. So this basically shows this pathway whereby we have a disruption of a controlled condition. In this case, it's going to be the plasma osmolality. That's going to have specialty receptors that detect this. These are called osmoreceptors. They detect osmolality. And these are in the hypothalamus, which their effect is going to be on the principal cells, such that the principal cells are going to become more permeable to water. So we're going to increase the water resorption in this region. Okay, and that's going to allow us to bring our plasma osmolality back to normal. All right, so as I mentioned previously, concentrated urine and dilute urine are made by 2 similar but different pathways. So this pathway is going to show you the formation of dilute urine. Basically what happens with dilute urine, so you have low ADH levels, so low antidiuretic hormone, which means that we can go ahead and let some water out. So the kidneys are going to produce diluted urine and excrete the excess water. And we are going to have the solutes resorb in that thick ascending limb, but the water is not necessarily going to follow by osmosis during that thick ascending limb. And at this point, the fluid that enters into the distal convoluted tubule here is going to be more dilute than the blood plasma. So the water is going to be leaving the body. In this region, in the early distal convoluted tubules, they're not permeable to water. They're not regulated by ADH, because if they're not permeable in the first place, ADH can't help them become more permeable. And when ADH level is low, the principal cells of the late distal convoluted tubule are also impermeable to water. So the water is not going to be coming back out. The permeability of these cells is regulated by ADH levels. So without ADH there, water doesn't get resorbed. That means that the fluid becomes more dilute as it flows onward, and eventually it's going to drain into the renal pelvis with a concentration that can be pretty low. So this is dilute urine. This is what we're aiming for, right? We always want our urine to be nice and clear, meaning that we have enough water in our body. But if we don't have enough water in our body, then we start forming concentrated urine. Okay, and concentrated urine happens when ADH levels are high. So the kidneys are going to, again, create concentrated urine by conserving water. So more water is going to be resorbed from the tubular fluid into the interstitial fluid, leaving the solute concentration of higher. This is the wrong one. Show me the next one. Here we go. Which is going to leave the concentration of urine high. So I apologize. This image is the same as the one previously. It should be in your PowerPoint presentation. You should pull out the other image, which should be the concentrated urine here. I apologize. I'll make that switch, but you should do that at home as well. So basically what happens at that point is that we're going to have more water coming across the collecting ducts. So therefore, we can have more resorption of water, but that's going to result in more urea recycling, which means that we're going to have a buildup of urea in the renal medulla, which will then end up in the urine. All right, so here's showing the countercurrent multiplication of concentrated urine. So I do have one of the concentrated urine slides. So essentially what happens, we have these long loops of Henle that are those juxtamedullary nephrons. They're really long ones that go all the way down through the renal pyramids. And we have symporters in one region, in the ascending limb, that's going to allow a buildup of sodium and chloride in the medulla. And that means since we are going to have this, we also have countercurrent flow, which means that we're going to have an osmotic gradient in the renal medulla. So we're going to end up with water being resorbed here, and then water also being resorbed there. So that means that here basically we're also showing what's happening in the blood supply. So here's the blood, and here's the urine. And since we are entering into a higher concentration, the water is going to flow out. Here, we are going to have sodium and chloride flowing out here, and then when we go back through, water is going to flow out through this entire section too, resulting in concentrated urine. And as the water is flowing out, it's going to get picked up. We also have the blood supply here is going to be losing water and picking up sodium chloride in this region, and then losing sodium chloride and picking up water in that region. So it's basically the opposite of what's happening in the urine. Does that make sense? So as we're traveling down into the more concentrated space, the water flows out. Right? And then as we're traveling back up, the sodium and the chloride flow out. And then as we travel back down again, the water flows out again, meaning that we end up with very concentrated urine. Okay. I'm not going to talk about evolution. Urine storage. Storage is important, though. So urine is basically going to drain into the minor calyces. The minor calyces join together to become major calyces. That's going to form the renal pelvis. And then that renal pelvis is going to drain into the urinary bladder and then out of the body itself by way of the urethra. Then development of the urinary tract. It looks like that is the end of urinary system. Oh, I guess what we are missing here is this last image. You guys have it on the screen. Here? All right. So we're going to have ruga inside a mucosal layer, and the ruga is going to allow us to expand, right? We also saw that when we looked at the stomach. We see it in the intestines. Here we have the ureters, which are coming down from the kidneys. They enter into the urinary bladder through these ureteral openings. Again, we have a little triangle region called the trigoin region, which is going to be where we're looking at the ureteral openings across and then down to the internal urethral orifice. As I mentioned previously, just like the process of defecation, they have two sphincters, right? We have the internal urethral sphincter and the external urethral sphincter, one of which is under voluntary and the other is under involuntary. So involuntary is going to be that internal urethral sphincter, and once that opens up, that gives you the absolute urge to urinate. And then that internal urethral sphincter is the one that's underneath our own control. And that's going to allow us, if we do relax it, to relax the urethra, allowing the discharge of urine from the body. That's called micturition, when the urine is expelled from the urinary bladder. And then last but not least, we have an image of the male and female urinary bladders. The urethras. As I mentioned previously, that the male has different sections. So the male is going to have mixture of both seminal fluid and urine, can both go down the same pathway. It's the same exit for the body. Whereas ladies, we are going to have separate reproductive holes, essentially, so our ureter does not connect with our reproductive pathways. That said, we have one section of the ureter. We just have the urethra. We just have the urethra. But men have the prostatic urethra, the intermediate urethra, and the spongy urethra. So that makes them less susceptible to urinary tract infections, but it does mean that they have much more complicated piping in this region because it also connects to their reproductive tracts. All right, so that brings us on to fluid homeostasis, which I hope I can kick through in 20 minutes. We will see. So fluid homeostasis basically refers to the body fluid. Body fluid is going to be about 50% of your body. The other 50 or 45% is going to be solids. Water is split up into two major things, the intracellular fluid and the extracellular fluid. Intracellular fluid is about 2/3 of that. 1/3 of that is going to be extracellular fluid. And when we split up our extracellular fluid, we have two different types of extracellular fluid, namely blood plasma and interstitial fluid, which is the fluid that bathes the cells. And this is a very rudimentary drawing, basically depicting exchange. We have exchange of gases, we have exchange of waste, we have crosstalk of these fluids, right? These fluids are constantly leaking from the blood plasma out into the interstitial fluid, from the interstitial fluid into the tissue cells, right, or the cytoplasmic fluid. So we're constantly having water exchanged as well as fluid exchanged. This is talking about, the next image is talking about water gain, which is water loss. So where do we get our water from? We get it from the process of metabolism, right, or dehydration synthesis. We get it from the food that we ingest and the liquids that we ingest. And then we lose water in two different mechanisms, sensible and insensible water loss. Sensible water loss is water loss that you can feel. For example, the process of urination, so through your kidneys or your GI tract. And insensible water loss is water loss that you cannot feel, like the process of respiration or the process of sweating, so through your skin or from your lungs. All right, so we want to make sure that we regulate our water concentration so that we don't have more water lost than water gained, which would lead us to the process of dehydration. Dehydration has three major effects. One, it decreases your blood volume. You have less water in your body. It also decreases your saliva, less water in your body. But the liquids in your body are going to have an increased osmolarity. So your blood is not only less of it, it's thicker, if that makes sense. We have an increased blood osmolarity. So the decreased flow of saliva makes you have that thirst center because you have a dry mouth, and pharynx makes you thirsty. The hypothalamus is involved when your blood osmolarity increases, which stimulates the thirst response. And a decrease in blood volume, resulting in decreased blood pressure, resulting in angiotensin II, also stimulates the thirst center. All of these are going to increase your thirst. Hopefully that's going to increase your water intake, which will return your body to normal levels. So this is normally how we maintain the proper amount of water in our body. Now there is a moment when we can get too far, when we have to make sure that we're not bringing in pure water, where we're going to have something like an IV that's going to also have electrolytes in there as well if we end up with too far into dehydration. All right, so under normal conditions, the fluid loss is adjusted for by ADH, ANP, angiotensin II, and aldosterone. These are all going to play a role in regulation of your water loss, which is going to result in regulation of your urination as well. Why am I looking at two different slides here? All right, so you guys missed one then? Yeah, you guys missed one. All right, so this is also talking about another factor for water balance, where it has to do with your salt concentrations, right? Because really what you're looking at when you're looking at percentage of water is how much free solute is in the water. So we're going to be looking at our salt concentration. And if we have an increase in our salt intake, that increases our plasma concentration of sodium and chloride, which increases the osmosis of water. Remember, water will always follow sodium and chloride, so it'll come out of the interstitial fluid and into the blood, increasing the blood volume. When blood volume increases, that increases the stretching of the atria of the heart, and that is going to result in an increase in ANP. On the flip side of this, we're also going to have less renin produced by the juxtaglomerular cells, and decreased formation of angiotensin II, which decreases the release of aldosterone, and all of that's going to result in increased glomerular filtration rate. These three things together are going to reduce the resorption of sodium chloride by the kidneys, which means we're going to increase the loss of sodium and chloride in the urine, which means that water is going to follow, right? Water is going to follow those solutes, which will then result in water loss in the urine and therefore decreased blood volume. All right. Now, this is an issue where we might end up, so when we're talking about water intoxication, where sometimes it's just too late to drink nothing but water, where at some point you really are going to have to give this patient some sodium or some electrolytes as well as the water. And this would be an example of when, if we have severe blood loss, severe sweating, vomiting, diarrhea, etc. So what we've lost is not just water. We've lost electrolytes as well. So that means that we've lost solutes in that water, such as the sodium. So we have a decreased sodium concentration or hyponatremia, which means that decreases the osmolarity of our interstitial fluid. And that means that the water is going to be pulled from the interstitial fluid into the intracellular fluid, causing the cells to swell up. Right? So this would be a time frame when you don't want to give this patient pure water. Even though you're like, wait, they've just lost a lot of volume of blood. We should really be making sure that they get rehydrated. what you should be doing is making sure that they get all of the constituents of the plasma back, not just the water, but also the sodium and the chloride, et cetera, which is why we'll give them a specialty cocktail for an IV for an individual who is suffering from severe hemorrhage or something like that. Otherwise, you can result in convulsions, coma, and death from water intoxication. All right, so the next slide is going to show us the different concentrations of the different major compounds in our body in blood plasma, interstitial fluid, and intracellular fluid, including things like sodium, potassium, calcium, magnesium, so all these different ions. And I don't need you to know the difference. I'm never going to ask you, is it higher in the intracellular fluid? Is potassium going to be lowest in the blood plasma, et cetera? But what I want you to see is that these are different. So the concentration of the ions that are in your blood is different from the concentration of the ions that are in your interstitial fluid. It's different from the concentration of the ions that are in your intracellular fluid. And that is going to help us maintain a gradient, whereby we can pull things in or push things out as necessary. And a lot of this is going to be maintained. Here's our sodium-potassium pump, right? Right, we're going to pump sodium out, pump potassium in. In that way, our intracellular fluid is going to have more potassium in it than any of the rest of them, and the sodium is going to be lowest because we're constantly pumping that sodium out, right? So really the take-home here is that the exchange of fluids is more than just the exchange of water. It's the exchange of the electrolytes, et cetera, and things are going to the cells that the cells need and away from the cells that the cells are pumping out, basically. And this talks about all of the different ions, potassium, chloride, bicarbonate, etc. I'm not going to read all of this off to you. We have multiple different types of buffer systems for our acid-base balance. So our body is constantly trying to maintain homeostatic conditions. One of those is going to be the pH. The pH is supposed to be approximately 7.4. So in order to maintain the pH, we have specialty buffer systems. We have three major types of buffer systems. The first is going to be a protein buffer system. The second that we're going to talk about is an acid-base buffer system, and then the bicarbonate buffer system. Come on. So the first one is going to be here. This is the protein buffer system. So in this way, we can see that, and this is going to be one of the most abundant ones in the body because we have a lot of amino acids throughout the body, but you can see that the proteins are able to either provide a free hydrogen if the pH rises too high, which means that it becomes basic, so the hydrogen is going to help bring it back down. So they can provide a free hydrogen by reducing here, by losing this H from the COOH, or if the pH drops too far, meaning it's acidic, we can absorb some of those hydrogen ions on the amino group and pull that in. Okay, so this is a nice protein buffer system in that we can release hydrogen if we need them or pull hydrogen in if we have too much of it. We also have hemoglobin as a buffer for carbonic acid. So hemoglobin is a really good example of a protein buffer system. So hemoglobin is able to pick up the carbonic acid and hold on to it, so it's able to hold on to this hydrogen ion that's released from when carbonic acid becomes bicarbonate. And then that hydrogen ion can get picked up by reduced hemoglobin as it's releasing the oxygen to the cells. So it can do two things, right? It's delivering the oxygen, and then after it delivers the oxygen, it's going to pick up the free hydrogen ion that comes from the conversion of carbon dioxide into carbonic acid and from carbonic acid into bicarbonate. So in that way, we're able to take this hydrogen ion and pull it out of solution so we don't have a drop in pH, which would be associated with an increase in hydrogen ions. Right? And so if the pH rises, then the hydrogen ion and the bicarbonate ion, I'm sorry, this is the next set. So this is the third one. This is going to be the bicarbonate carbonic acid buffer system, which is one of the most common ones in the blood supply. And basically what happens is if the pH rises, that means we have extra hydrogen ions in solution. And bicarbonate, which is a weak base, can pick up that hydrogen ion and create carbonic acid, which is a weak acid. Now, if we have a pH dropping, this weak acid can release this hydrogen ion, becoming bicarbonate, and putting the hydrogen ion back in. Right, so we'll be converting it back into a weak base. All right, and so this is a really important regulator of pH in both the blood cells, also in the tubular fluids, etc. We also have the phosphate buffer system. The phosphate buffer system looks like this. Here we go. So the phosphate buffer system is going to involve dihydrogen phosphate and hydrogen ions. So instead of picking up, in this case we were previously picking up the hydrogen ions, we're just releasing the hydrogen ions. This one also will allow us to pick up the hydroxide ions. The hydroxide ions, relative to the hydrogen ions, are basically what pH is. And so if we end up with more hydroxide ions in solution, then we end up more basic. So if the pH rises, it gets too basic, we can take hydroxide ions out of solution and convert them using dihydrogen phosphate, which is a weak acid, into monohydrogen phosphate, which is a weak base, and releasing water. And if the pH falls too far, we can use this hydrogen ion, convert it with that monohydrogen phosphate to become dihydrogen phosphate. So we can pull that hydrogen ion out of solution. So this is going to allow us to maintain the pH using the phosphate system. And this is kind of unique to the others because it's going to allow us to pick up the hydrogen ions out of solution as well as the hydrogen ions out of solution. All right, so the next thing that we're going to talk about is the exhalation of carbon dioxide and how that is going to be responsible for the concentrations of pH in the body. So when carbon dioxide is exhaled, the blood pH rises. And when carbon dioxide is held in the body, the blood pH falls. Why? Because carbon dioxide can get converted to carbonic acid, and carbonic acid is obviously an acid, which brings our pH down, right? So when we are getting rid of bicarbonate to carbonic acid to expiring carbon dioxide, that means that our pH is rising because we're getting rid of what can become that acid, right? We're getting rid of the fuel which can drive the creation of that acid. And so this is constantly happening during the process of respiration where we're off-gassing carbon dioxide, and then when we are getting that carbon dioxide from the tissues to be delivered to the hemoglobin or whatnot, it can, in the plasma, become bicarbonate, then get converted to carbonic acid, and then when we respire in the lungs, off-gas is carbon dioxide. Okay, so we're constantly having small fluctuations in our pH just based on how much carbon dioxide we're retaining at that moment, which is really just how much we're respiring relative to how much our tissues are going to be needing. Right, so the rate of breathing is affected by the pH of the body fluids, because if your pH ends up too low, you probably should be breathing more. So you might end up having to increase your body breathing rate because you have too much acid in your bloodstream, so you want to be off-gassing some carbon dioxide. On the opposite side, if you do a quick hyperventilation where you breathe really quickly for a short period of time, and then you can hold your breath for longer without having to that inspiration reflex because you've already off-gassed the carbon dioxide, so your pH has risen, and so you have a little bit of time for that pH falls again. All right. So the next one is going to talk about mechanisms that maintain the pH of body fluids. So just like anything, this is going to be some sort of stimulus comes in that disrupts a controlled condition. In this case, the controlled condition is blood pH. And so if we end up with an increase in hydrogen ion concentration, it means we're decreasing our blood pH, right? Our blood's becoming more acidic. And we have specialty receptors in the medulla oblongata and also in the aortic and carotid bodies. We talked about the aortic arch receptors and the carotid body receptors previously when we talked about baroreceptors and stretch receptors, but in this case they also have chemoreceptors. And that's going to send information to the control center, which is the dorsal respiratory group, or the DRG. And that's going to have effectors in terms of the forcing the diaphragm to contract more forcefully and frequently so the carbon dioxide is exhaled. Eventually, we're going to end up with less carbon dioxide in our body, which means that we have less H2CO3 forming, which means that our blood pH is going to increase because we have less hydrogen ions because we've off-gassed some of them, right? And that's going to allow us to, because we've off-gassed carbon dioxide, which allows us to change our pH. So now our pH is going to come back up and return back to normal, thus restoring our controlled condition. Oh, all right. Well, it looks like I'm almost on time here. What else am I missing? So we have some issues where we can have blood pH problems. So, for example, we can have blood pH that is acidic. It's called acidosis. We can have blood pH that's alkaline, called alkalosis. And there's compensatory mechanisms on both sides that return your pH back to normal. And what do I mean by compensation? All right, so we can have respiratory issues, where we have respiratory alkalosis or acidosis, or we can have metabolic issues. And if we have a problem on the respiratory side, we can compensate by metabolism a little bit. And if we have a problem on the metabolic side, we can compensate on the respiratory side a little bit. Okay? And so basically what's happening is you have two different things happening. You have the respiratory rate, which is saying how much oxygen and carbon dioxide are coming in and out of the body. And then you have the metabolic rate, which tells you the oxygen consumption and needs of the cells. And these are supposed to be in concert, but if they end up a little bit of fluctuating, then you can increase one to meet the demands of the other, or vice versa. All right, we don't really talk about aging on electrolyte balance, although we did talk about dehydration in older adults as well. Okay, so that brings us to the end of our lecture. Wow, I really did auctioneer style it. Do you guys at home have any questions? Go ahead and just unmute and ask them. Same with you guys in the lecture. Have questions on anything? No? All right, I'm going to go ahead and stop this. Thank you so much for listening to our review session.